Thank you, Rucheng, so much for inviting me, introducing me, and thank you, Bruno. This was a very inspiring talk. And now, prompted by Bruno, I also have to show off my international credentials by saying that I was born and studied in Bulgaria, and then I studied some more in Canada, and now I'm here in the US in California, and hopefully go to other places too later on. So I will, from this broad overview that Bruno gave us, I will now focus into finding the right antibodies. So a lot of our work with electron microscopy depends on antibody labeling to be able to localize, find the location, see the distribution changes with time in different proteins. And we use array tomography, which in a way is basically one variant of post-embedding EM. So looking for antibodies for array tomography also has the side benefit of finding antibodies that generally would work for other applications of post-embedding electron microscopy. Well, I would just, I guess Bruno went over this, but I will quickly remind about our technique that we're using. With array tomography, you would fix your sample, dissect and embed in resin, and then cut serial sections, ultra thin sections, or sometimes semi-thin sections. And then we usually pick them up on cover slips, which works great for immunostaining and then fluorescence imaging. So the serial sections, we immunostain them usually with a combination of 3 different antibodies, and there is also DAPI present to stain the nuclei and give us the cellular context. Then they're imaged with fluorescence microscope, and after that, the antibodies can actually be eluted. So we use a high pH solution that can remove the antibodies, and this allows another set of antibodies to be applied and imaged again exactly in the same location, and thus acquire quite extensive molecular information from our sample. And then whenever the fluorescence imaging is done, and if the tissue is prepared the right way to be able to be used for electron microscopy, then one can post-stain it with heavy metals and image in the scanning electron microscope and obtain the ultrastructure from exactly the same area where the immunofluorescence was obtained. Now, this is all great, but it relies heavily on antibodies. If you don't have the right antibodies, it's not going to work. And finding the right antibodies can be really painful, long, expensive process. One big reason for this is that all the commercial antibodies are really not tested for this application for tissue that is usually dehydrated, embedded in resin. There's a lot of changes that happen. So there is absolutely no guarantee that antibodies that work great in other applications will work in our application. So one old and great way of checking antibodies for electron microscopy is actually using light microscopy on exactly the same tissue that one would later do electron microscopy. And here I am showing you the example of the validation of the GABA antibody. So GABA is a small molecule, a neurotransmitter used by the majority of inhibitory neurons in our nervous systems. And this is a really useful antibody. I think it's probably one of the most used in neuroscience. It is great because it works for electron microscopy, like for tissue that is prepared for very standard electron microscopy. So fixed with osmium, embedded in EPON, all that things. And originally, when it was first developed, Peter Somogyi showed in his article how it looks on semi-thin sections from cerebellum, which is actually enough to make one convinced that it works for GABA because of its very specific distribution. So one can see the Purkinje cells that are stained with GABA, and also other smaller cells, which are other interneurons, and they are very immune positive for GABA. And this such a view at the light microscope can help really distinguish an antibody that works or doesn't work, and save a lot of trouble before going to electron microscopy. So exactly this antibody on this same tissue now viewed with GABA immunolabeling shows that it works for GABA immunogold 2. Here you see two terminals that are synaptic terminals that are on a pyramidal dendrite, and they're labeled with immunogold. And the same tissue preparation also works for many neuropeptide antibodies, like in this case, CCK is localized to exactly the same neuron that also is labeled with GABA. And this is a known fact that some of the interneurons also are labeled with different neuropeptides. So there are some antibodies that work great for this traditionally prepared electron microscopy tissue that preserves the ultrastructure really well. But these are mostly just small molecules like GABA, glutamate, or neuropeptides. There's very, very few other antibodies that will work under these good conditions for ultrastructure. And they require a bunch of pre-treatments to be able to get a good immunostaining with these antibodies. For immuno-EM, a lot more useful is tissue that is embedded in low-acryl. And in this case, osmium is omitted, and the tissue is prepared with using freeze substitution and then embedded in low-acryl HM20. And now there's a lot more antibodies that work under these conditions, including these antibodies that work with the osmium tissue, but now the repertoire of antibodies is much more expanded. There's less pre-treatments that one requires, and the ultrastructure is also similarly quite good and sufficient to identify features, many ultrastructural features like synapses in this case that we're interested in. And you can see that immunolabeling for different synaptic molecules now works really well, but it doesn't in the case with EPOM embedded and osmium post-physics tissue. Even though there's a lot more antibodies that work in this case, it's still a pain trying to identify them. So we try to do this mostly at the light level because it saves a lot of time, it is faster, and there's a lot more readouts that one can apply to judge whether an antibody works well or not. So it becomes much easier to compare the immunolabeling with other antibodies that have similar distribution, or then check whether it would label locations where it is not supposed to label. So use it together with an antibody against an spatially exclusive antigens, or check background nuclear staining. So there's a lot more possibilities and it's much more efficient. Now, usually the way our strategy works in this case is, well, first one needs to develop a set of antibodies, like a marker antibodies that will help you identify in your tissue the different compartments. So then one can judge where the antibody that you're testing is actually distributed. So a lot of our work is with synapses in the mammalian cortex, and that's what I'll show you as a first example. We have developed the panels to mark different compartments. And of course, synapsin is one very ubiquitous marker for any kind of synapse in the cortex, not everywhere, but in the cortex. Synapsin is an excitatory synapses and inhibitory synapses. And then when we want to find specific synaptic types or synaptic compartments like presynaptic versus postsynaptic, then there are different set of other abundant markers that can help us distinguish between the presynaptic part of an excitatory synapse versus postsynaptic, and the same for an inhibitory synapse, presynaptic versus postsynaptic. So here is just one example of us trying to validate a neuroligin 2 antibody neuroligin 2 is known to be associated with inhibitory synapses. So here is a very small volume, which is labeled with synapsin. And then we can apply the different markers to differentiate between different types of synapses. VGLUT1 in red would label the majority of excitatory synapses in cortex, about 80%. Then the remainder are the synapses in yellow, which have VGLUT2, and the inhibitory synapses are in blue. And then when we add the neuroligin 2, you can see that it associates mostly with the inhibitory synapses, and that's where it's supposed to be. So we're fine with this antibody. And another example is looking at myelinated axons. In a similar way, one can identify molecules that are abundant in different compartments of the myelinated axons, and then try to validate the antibody against where it's known to be distributed. So looking at potassium channels are known to be next to the node. So the node of the myelinated axons is the part where there's no myelin and where the sodium channels are distributed. Now the potassium channels are known to be right next to it. And as you can see here in these three different sections, we've labeled myelin with MBP, which is very abundant. So this is the myelinated portion of the axon. Here where you see no myelin is the node. And on serial sections, we can see that this specific potassium channel antibody, the labeling is very consistent and it's right next to the node where we would expect it. So that's another one that's passed the test. One can also use not just antibodies as markers, but other features or other markers. It turns out DAPI actually labels mitochondria well too. So here are the nuclei labeled, brightly labeled with DAPI. Here they're dendrites. And these little lines that are consistent from section to section are actually mitochondria. And then when testing an antibody for mitochondria, we can see that it's right on top of the mitochondria. So normally nowadays we use DAPI to stain the nuclei, but it turns out that the very first use of DAPI was actually for detecting mitochondria. And it's just, you have to crank the exposure to see it because there's much less DNA in mitochondria. Okay, that's another example of testing different antibodies for mitochondria. And again, here you can see the mitochondria labeled with DAPI and the antibodies right on top. So these are other antibodies that pass the test. Okay, so this way of screening is really useful for when you have a small number of antibodies. But what about actually trying to use light level screening for big projects or during antibody development? So we reasoned that the reason that it's so hard to find good commercial antibodies that work for post-embedding conditions, it's one of the big reasons is probably because that's not how the antibodies were selected, right? During development of antibodies, different screens are applied to select the antibodies that work. So if you screen for antibodies that work for Western blots, that's what you'll get. If you screen for antibodies that work for immunohistochemistry, that's what you'll get. So we probably need to screen for antibodies that work specifically for post-embedding to get those antibodies. But then we'll need an easier way to screen. So the first thing to simplify is the resin. Using low-acryl is complicated. It works great for ultrastructure and antigenicity, but it's not an easy protocol. It takes quite a bit of time to embed, and it's quite toxic. LR White, on the other hand, it's very easy. It comes in the bottle, it's ready to use. It's just a very simple recipe. It's fast and it's less toxic, and the antigenicity is great. We found from our experience that about 75% of antibodies that work on LR White would work on low-acryl. So LR White is actually a good resin to use to screen for antibodies that would eventually work for low-acryl. And here is an example where a tissue mouse cortex embedded in LR White versus in low-acryl HM20, labeled with different synaptic molecules. And it's basically the labeling pattern is indistinguishable in most cases. There are some, like as I mentioned, about a quarter of the cases, you would end up with antibodies that could not work with lowicril. This was an antibody against VGLUT2 that labels only some of the excitatory synapses, and with LRYT you see nice colocalization in some synaptic codons, but we don't see that with lowicril. And then one should also take into account the effect of the fixative. Some antibodies actually require glutaraldehyde. So the GABA antibody that I showed you, it gives spectacular results. It needs glutaraldehyde, and with no glutaraldehyde, you see no magenta label here. And myelin, the antibody would work without glutaraldehyde, but the structure of myelin is not well preserved. So you see here, it's much more like fragmented compared to the nice continuous look with glutaraldehyde and with lowicril. Okay, so that's for the resin. We figured out LR-white is much easier to test antibodies. What about the actual test? Well, using brain sections, in our case, we're interested in synapses, is actually quite complicated because you have such a complicated environment, and it's hard to judge depending on, some antibodies will be easy, but it's actually quite hard to judge if an antibody works or not. So we need something much more simplified. And that's where we came up with this transfected COS cell screen, where COS cells were transfected with the target protein for the antibody, and also at the same time with GFP. So you end up with cells that are green because they express GFP, but you also know that they express the target protein. Now, when you add the antibody, if it works, you would see colloquial labeling of only the cells that express GFP and the target protein. So colocalization here shown in white. If the antibody doesn't label, well, you only see green. If the antibody sticks to everything, labels indiscriminately, then you see some colocalization, but also a lot of magenta-only antibody. So together with UC Davis, with NeuroMab, which is a great place where they develop antibodies together with NIH, and the goal is to develop high quality antibodies at a low cost. And we decided to implement the array tomography test within their normal production of antibodies. So normally they would screen their anti-sera coming from mice using two different ELISA assays, one on COS cells, similar to what I showed you, the similar strategy, but just fixed with formaldehyde COS cells expressing the target protein, and also protein ELISA. And then they would choose the clones that scored the highest on both assays, and then test them on a number of different tests, which are the most useful for most people, like brain immunoblock, brain immunohistochemistry, or again, COS cells. And the top candidates from these assays will become the final antibody that is produced and distributed. Well, now the idea is, let's incorporate also the array tomography, the screening on sections embedded in resin. So now the top candidates after these primary assays, and usually it's 96 to 144 candidates at this stage, are also tested with this proxy screen. So now the proxy screen, as I explained, is COS cells that are transfected with the protein of interest. But now it is processed for post-embedding. So the cells are fixed and embedded in LR white resin. Then they're centrifuged in the pellet. So that's the pellet that's embedded. And the pellet is then sectioned into semi-thin sections that are put in 96 well plates, and then stained just the same way that what we would use it for array tomography. And then from here, using this readout that I showed you, green cells, do they label with the antibody or not? The top candidates are selected, and then only they are tested on brain sections, which is the harder part of testing. And then the top candidates are now the candidates that can be used for array tomography and for post-embedding. So here is an example of screening for Gefrin. These are these COS cells that were pelleted, fixed, and embedded in LR white and then sectioned. You can see the GFP expression in some of the cells, and in this specific clone of the antibody that works, see that the brightly green cells also labeled with the antibody and not much else is labeled, but there's a lot more cells. This is the DAPI staining, and here are all the channels. And examples of antibodies that were not selected. This one clone did label the cells that express the target, but there was very high background. And this one just labeled everything. So these were rejected. Now, these clones afterwards, then the top clones are also tested on brain sections. And now you can see how it performs in the brain. Here it is used with another together with an antibody to GAD, and GAD will label the presynaptic boton, and Gefrin would label the postsynaptic part. And if you focus on this specific combination of the two antibodies, which tell us there is a synapse there because the presynaptic label and the postsynaptic label are opposed, and it's consistent from section to section. This is this same part followed on 3 consecutive sections to convince us that the staining is real. And this same antibody also works for immunogold EM. Now, this is actually a section embedded with low-acryl HM20, and you can see the gold particles here on the inhibitory synapse. Here I've color-coded it to make it easier because that's the inhibitory synapse, presynaptic boton, postsynaptic density. And there are several excitatory synapses in the field of view, but there's no label there, so it works. And here is another example with a screen for Homer1. That's again done with Neuromab. And Homer1 is known to be at the postsynaptic density now of excitatory synapses. So here we also did some more extensive characterization of all the different clones. Like I think here there were 144 clones that were selected to actually convince ourselves that the proxy screen really works. And you can see that CBS is the proxy screen with the cell-based screen, so the COS cells. So the highest scoring COS cells have the most number of clones that would score high in brain evaluated with brain array tomography, while the clones that didn't score high with the screen, there was hardly anything that worked for array tomography. And the scores on the cell-based screen versus the brain screen correlate pretty nicely. So if they score high on the cell-based screen, they score high on the real application in brain. So this gives us a really nice predictive screen that can be used during screening of a large number of antibodies, because I forgot if I mentioned, but we didn't find any correlation on the other applications with the performance of the antibody with array tomography on post-embedding. So even if it's immunohistochemistry on brain sections, which is pretty close, but it still has no correlation with the performance of the antibody in post-embedding conditions. So here is just the same. Here the antibody, because it's known to be at excitatory synapses, it's tested together with PSD95, which is another protein distributed on the postsynaptic side of excitatory synapses. And again, looked at 3 serial sections. You can see that they co-localize quite well. These are 2 different clones. And then similarly to Gefrin, we applied it afterwards for immunogold labeling on low-acryl embedded tissue, and it works both in the mouse cortex, here's the postsynaptic density and some gold, and also on human tissue. This project, the antibodies that we tested in this way, is actually available on the Neuromap website, where you can see these are the tested with the different conditions with LR white, and then for some antibodies we continue and tested in low-acryl and in human, but this information is available now on the website, and it's helping us a lot to find the good antibodies. And I think it's time for me to start finishing. So I would like to thank all the people that participated in this project, and the lab that I'm in, the Madison lab at Stanford, Dan Madison, Mark and Mariana, a lot of the work in the development of the tissue for electron microscopy, the lowical tissue, is done at the University of North Carolina and Richard Weinberg. UC Davis is where we did the screening of the antibodies, and it's now part of their screen for developing new antibodies. And then also Stephen, Jenna, Forrest, and John from the Allen Institute. And then from UCSF, Bill Seal and Edward Chang for the human tissue that we used, and Guillermo and Anish for the computational algorithms that we used for evaluating antibodies. Thank you all, and see you later in Gather Town.